especially any new members that we happen to have, and old members, of course, welcome back. And just a quick note, if you would all please put yourselves onto mute. That way we won't hear any dogs barking or whatever in the background. And what we'll do is while we're having the talk, we'll have people add questions in the chat room. So if you have any questions, just type it into the chat room. Hopefully you know how to do that. If you don't, I guess raise your hand and we'll walk you through it. But anyway, thank you very much for coming. And it's my pleasure to introduce our speaker for tonight. And her name is Dr. Diana Northam. I've known Diana for quite a while now. We are both members of the National Speleological Society. And we meet frequently at the meetings, especially in the biology section. Diana earned her Ph.D. in biology from the University of New Mexico. She's been studying cave life since about 1984, a long time ago. And what's interesting is that she and her colleagues have founded a group called SLIME, which I think is a really cool name. And it stands for the Subsurface Life in Mineral Environments. And Diana has been caving and researching in Lechuguilla Cave in New Mexico, which is probably the most awesome cave in the whole planet. In my opinion, it's fantastic. And I'm very envious of her dry caving. I call it dry caving. And
it is dry caving.
Dry caving. So with this, I will turn it over to Diana, Dr. Northam, and we'll be happy to hear your talk about cave microbes. Thank you.
And just to start it off with a funny story about Lechuguilla. So I am terrified of heights, and Lechuguilla is nothing but pits. And when I was deciding what to do my PhD on, my PhD advisor, I was actually a faculty member in another department. And he was like, I gave him all these projects and he's like, no, we're going to do Lechuguilla. We can get money for it. And we got a National Science Foundation grant. And I got everybody to sing for me as I went up the drops, because when the top started to come in and the bottom faded out, I knew I was getting near the top of Boulder Falls. So that was how I dealt with that. So I am now going to share my screen. Okay, does that work for everybody? I see, okay, Paris is nodding, so I'll take that as a yes. All right, so I'm going to talk about something that we've been working on for the last decade, which is basically the idea that it's not the obvious that's actually going to make some of the interesting discoveries that might help us with knowing how to look for life on other planets or moons. It's actually a lot of what's hidden within the secondary mineral deposits. And we're concentrating on lava caves because there's so many of these deposits. And I'm going to present some of the work that a undergraduate senior honors thesis, Joe Medley, did. And he lived, by the way, in Nashville, Tennessee for a couple of years, and also part of a Braille project. And I'll tell you more about that as we go on. So I'm going to start with just a little bit about why caves are so different than surface environments. I'm going to ask you, unmute yourself. And if you've ever been in a cave that has no artificial light, where you had to bring the light, just call out yes, so I can get an idea of how many people. So unmute yourself.
Yes.
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 Yes. Okay, and Harris is going now. Okay, so this is for Harris, okay? All right, so it's dark. And one of the things I've discovered with students is they don't realize how dark it is, that you can't see your hand in front of your face if all the lights are out. The other thing that's important for life is when you're in the deep cave zone, where the cave is adjusted and isn't influenced by the outside as much, it is 100% relative humidity. And the temperatures are relatively constant. And one of the things I learned was that they are that's really a term, relative, because I now work with a German cave microclimate expert, and he's shown me that there are fluctuations more than I thought there was. They're not much, but they are there. For a microbe or a cave invertebrate, like some of the ones that maybe Jill studied, she studied crustaceans, at least in the caves I work in, there's much less to eat. But if you're a microbe, what there is a lot of what's called reduced energy sources. So within the rocks that you see in this picture, there are forms of manganese or iron or sulfur that when the microbes go in and transform them to an oxidized version, they gain energy. So this is where they get, I call them rock eaters. And so they're able to gain their energy from the very rocks that you see. The other thing that's important is there's also minimal weathering. Makes a big difference. You're not completely, the community is not getting washed away periodically. So reasons to study this. So because the caves have a lot, they have no light, and they don't have a lot of variation, they make really good geomicrobiological laboratories. So the intersection between geology and biology, because you can take a lot of variables off the table. They are also a really good analog for thinking about or characterizing what is life likely to be like on another planet or a moon in our solar system or elsewhere. Penny Boston, who's one of my colleagues and works for NASA, has basically had a long history of investigating this question. And it's basically a function of, it goes all the way back to 1992, her paper then, and actually there are earlier evidences from other people of pictures showing that there are lava caves on places like Mars, the moon, and other places. And so this has become an even hotter topic for NASA and other astrobiology agencies to look for, okay, can we find life in the subsurface of Mars or elsewhere? And this just shows you that Venus and Io and the moon have lava caves on them that would be possible good investigation targets. So here's the rub. When I go into a lava cave, for instance, what you're seeing are on the left, a lava cave in Hawaii, and on the right, a lava cave in the Azorian Islands. So who can tell me where the Azorian Islands are? Any takers? You got to unmute yourself, Jill. Nope, still
I'm
not.
sorry.
Okay.
It was either my computer or yours, but your question was a little muddy. So could you repeat the question, please?
So who can tell me where the Azorian Islands are?
In the middle of the Atlantic.
Yes, they're
Somewhere.
1,000 kilometers from the Portuguese border. So are you hearing me okay, or do I need to do, okay, it's all right. Okay, so what you're seeing in the Hawaiian cave, all this pink, those are microbial colonies. And each colony has literally millions of organisms in it. And this is a major portion. So this is really a big metropolis. This is the Tokyo of microbiology in this Hawaiian cave. So over here, this is an individual colony in a cave in the Azores, and they often are hydrophobic. They repel water, and that's what you're seeing are water droplets on this colony. But the issue is, what about something like these little fingers? I hope you can see my cursor moving back and forth here. These little fingers, polyps on here. One of the things I've discovered over the last 10, 12 years are you actually can find a lot of life in what looks like a secondary mineral in this cave. And so we're going for finding out what's in these less obviously microbial, more mineral in appearance. Because when an astronaut or a rover is on a moon or another planet, how are they going to know what to sample, which samples to choose to evaluate what life is present? Okay, so that's what we're going for. So, so we're going to look at some of the studies we've done over the last 20 years. These have been focused in Hawaii, that you see here on the left. This is a lava cave in Australia, and this is a lava cave in the Azorian Islands out in the middle of the Atlantic. So we're going to look at some of what's present there. So one of the reasons that I really like the Hawaiian caves, and I'm going to present you a lot of the Hawaiian stuff, is because, and this is Joe Medley, by the way, doing research in there. If you look at these walls, you'll see all those light colored, either white or yellow or tan. Those are secondary mineral deposits, mineral crusts on the walls that actually have a great deal of microbial life. And so we have on the big island of Hawaii studied a lot of caves over here on the eastern side. This is the wet side. Hilo gets between 3 and 4 meters of rain a year, versus down here in Ocean View, where I'm going to show you some of the work we've done. And this is where Kenneth and I actually have a Quonset hut that we go to for a couple months a year and do research in the caves. But down here, they get on the order of just 22 centimeters. So it's a lot drier, and Kona is known as the dry side. I'm also going to show you briefly a cave up on the slope, way up on the slopes of Mauna Loa here, and how different that is and what the potential is for us finding things. And somebody else is monitoring the chat, right, to see if there's anything I need to pay attention to. I saw a 1 there. So let me know if there's anything I need to answer. So these are the very obvious microbial in Hawaii. This is where we started with these. But then we discovered this. Do we have any geochemists in the group? Anybody who's interested in what the chemistry of some of the rocks is? If you are, then these were a gold mine. So this is actually a speleothem, it's only about 1 centimeter long, but that blue-green is a copper silicate. And this was just one of the most exciting finds, because I was actually with a cave rock mineralogist when we found this. And so this was really neat. But we started looking across all these caves, not just at the blue-green deposits, the turquoise, but at these sort of rusty red-orange deposits, which have a lot of iron in them. And also right here, you can see a stalagtite with a water drop on it, and there's goo on it, what we call ooze. And it looks like somebody took a jar of baby food and threw it against the wall of the cave. That's the consistency that you're seeing. And I first saw this in the Azores. I saw this in the Azores. These are iron hexagons on this rock that was in the middle of a tourist cave. So this across here is only on the order of 4 centimeters. This is really small, but there were all these amazing structures. So we started characterizing these and looking at different shapes. Is it like a mineral crust that is, I can't seem to stay in the picture. If that's bothering you, I can turn off my virtual background. Just let me know. Jill, maybe you can say like this if I need to turn off the background. So it's okay. All right. So the shapes can be knobby. They can be a flat mineral crust. They can look like a head of cauliflower, a small head of cauliflower on the wall. They can be little pimples or coralloids. So branch structures like you would see in a coral reef. And the textures can be flat or they can be bumpy. There can be textured rims, and we get different colors. And that's one of the things that we analyze. So we're going to look at some examples now. So these are the ones that I said are very widespread. We've seen this in caves, in lava caves all over the world. And they do have iron in them. This one is from Thurston's Lava Cave on the Big Island of Hawaii. And if you look at it with a scanning electron microscope, you can see what's inside. And so this is magnified several thousand times. And so to give you a sort of parameter, so this bar up here on the upper one and the same bar here on the lower one, both are 20 microns. One of my hairs is 100 microns in diameter. So this is 1/5 the width of one of my hairs. But you can see here's a filament. There's a whole lot of round microbes in here, a whole bunch on this ball. And then up here, you see biofilm. So biofilm is what you feel on your teeth in the morning, have to be before you brush them. You know how it feels sort of slimy? Well, microbes produce biofilm. So this is like the slime on your teeth. And for them, it's a protective film that prevents things from getting to them. So if you had these in a heart valve, for instance, and they had biofilm, when you took an antibiotic, it would not be destructive to the inner ones because they'd be protected by this biofilm. So we did a study. This is one of my husband's photos that he did with it. We used for an article we published in the journal Astrobiology. And we looked at those blue-green. And when you look inside them, all of these that you see that sort of look sort of fuzzy, those are all a group of bacteria called Actinobacteria that we get 2/3 of our naturally produced antibiotics. And the geologist I work with, Mike Ph.D, did an analysis of it and determined that they were actually a poorly crystalline mineral called chrysocolla. It is a copper silicate. And then if you look at the stringy things in here, these actually look like a woman's fishnet stocking. But this is a 2-micron bar. These things are really tiny. They are about 1/200th the width of one of your hairs in terms of their width. So these are really, really tiny, but we have found these in caves all over the world. And so one of the things we do is we take a sample with permission. And in this case, it's a privately owned cave, and we got permission from the owner. We took a small sample, not of one of these really pretty ones, and we extract the DNA. So just like when you send off a saliva sample to know your genealogy and have it sequenced, we take a small sample, we extract the DNA, we sequence it, and then we compare it to databases, and it tells us who's present. So what you're seeing, see how many of these names start with nitro? That means these guys use nitrogen as an energy source. In this case, or hydrogen, which you wouldn't think was really nutritious for you, and sulfur. And that's the thio that you see. So these guys are able to eat substances in the rock. And the other guys that are in here are able to withstand the toxic effects of the copper that's in here. And that's what this guy is. Cupria vitis is a copper resistant organism. It's able to withstand these high levels of copper. This is what it looks like inside. Lots of filaments, these really cute looking balls that we have no idea yet what are. But when you use an extra tool, so one of the things Mike Ph.D has on his scanning electron microscope And I could do this for hours, days, weeks on end, because you see all these amazing structures in here. When he applies the electron beam to this, he can basically tell what the elemental composition is. So for instance, this ball, which is up here, or down here, sorry, has silica in it, and it has copper, and it also has a mineral called vanadium, which is kind of amazing. I have not seen this in caves before I started working in the lava caves. And then when you look at some of the minerals present, you find another one, bismuth. And it tells you sort of, relatively speaking, how much is present. And it gives you ideas, what could these microbes be eating? So it's like somebody went through the supermarket and analyzed what was present. One of the things that I think is prettiest in the lava caves are these gypsum round spheres. So I call these spheroids. And they just adorn the walls and make it look absolutely fabulous. And you can see in the SEM, they have some really interesting mineral shapes. You wouldn't think I didn't think something like this would have minerals. But here's a scanning electron microscope that's down at 5 microns. So this is 1/20th the width of one of your hairs. And look at all these filaments and all these other microbial shapes. So this is one that has lots of microbes in it. And then one of my favorite things in these lava caves is what's called moon milk. This is my favorite for many reasons. And one of the big is back in the Middle Ages, basically people in Europe use these as a, they'd put it on wounds, and there are a whole bunch of those actinobacteria that I told you about. Those are able to kill the germs in the wound. And so this moon milk was a great source of these actinobacteria. And this is a fluffy one. A funny story is one of the geologists that I work with in these caves thinks that the way you tell what a mineral, well, he knows that a way to tell what minerals are there is you lick them. And I'm like, oh, Stefan, you don't want to do that. There are also bad microbes in there. This is a really bad idea. So if you are a geologist, don't do this. Here's what this one looks like inside. So you can see all these little filaments. So all of these are bacteria. Hold on one second. We have cats that are coming in going, is it dinner time? Is it dinner time? So you're hearing in the background a little bit of dinner preparation. And then here are those microbial colonies that I told you about before. So these are, and I'm going to put on my headset, which I don't have. I guess I'm not. These are the microbial colonies. And when you look closely at them, there are all these little round balls that have all these little spiky things. These are bacteria. And that's what you're seeing here is just lots of these. Now, here's that ooze I was telling you about that's all over the walls. It has those reticulated filaments I told you about that you can see here, and it has all kinds of other shapes of microbes in it. This stuff is actually really full of microbes, which is amazing to me. We're trying to study how this actually forms, because you don't, the kinds of caves that Jill goes into, you wouldn't see this because these are dry and it doesn't get washed away. So what Joe did with this for his senior thesis in college was he took all these different shapes and looked at them across the continuum, because it turns out these mineral crusts, except for the copper, don't have that many microbes. And then as you go along this continuum, you get more microbes. So ooze and these moon milk and these microbial mats have a whole lot. And so it's one of the ways that we help sort of select what are the best targets for an astronaut or a robot to select. Now, this looks really complicated, but what I want to tell you here, this is the new tree of life that was made just 4 years ago. And so what has happened? When I first started studying this back in 1987, all this, which is the bacteria up here, all these upper ones, there were 11 different phyla. There are now over 100, and all the red dots are ones that have never been grown and never been characterized. So look at all the new stuff we are finding. And they're only known from their genetic sequence so far. The other group, the archaea down here, are really important because it turns out they play a big role in cycling nitrogen. And nitrogen's important for all of life. And so that's really critical. And then we humans are way down here at the tip of this branch. So we're on the tree of life, we're somewhat insignificant. Okay, I'm gonna just make one point about this. I want you to look at the list of names. See how many of them say nitro something? All the nitro ones are ammonia oxidizers. So one of the big mysteries we're just encountering is, where's the ammonia coming from in these caves? And that's something we actually don't have any good idea about. And we're working with geologists to try to work that out. The other thing I'd like you to notice, these three are caves in New Mexico. These three are caves in Hawaii. Notice how different the color patterns are in these. So who lives in the caves on Hawaii is really pretty different than who lives in the caves in New Mexico. So geographically, you get a lot of difference. And part of that is because the makeup of the lava is different. This is, those were the archaea. These are the bacteria. And I've put a star by a group called Nitrous Fire. This is a really important nitrogen cycle bacterium. And it's present as a major part in all these different caves and several samples across these caves. So that's one of the things we're discovering is the microbes are sort of the base of the food web. They are transforming the minerals and making food for the rest of the thing. So now we're going to take a quick trip up Mauna Loa. So we are at 11,000 feet, which for those who do kilometers is probably around 3,000 or 4,000, 3,500 not kilometers, meters above the Earth's sea level. You're looking across at Mauna Kea, and you can see the observatory up on Mauna Kea. And one of the things that jumps out is there's no life up here. Notice there's not a green plant to be found. And so that source of carbon up there is not present. So these caves have even less to eat. And one of the things we discovered in this cave, you may remember I mentioned on that one elemental plot that vanadium was present. So having the ability to see small different things is really important in caves like this. So notice this where my cursor is in the upper left, this yellow, sort of yellow gold. It turns out that's vanadium oxide, which I would never have considered in a lava cave to be a, a home for microbes. But when we took a very small sample of this and looked at it in the scanning electron microscope, we saw life, all kinds of life in there. This happens to be gypsum, but this little thing that looks sort of like a turd is actually full of vanadium. So I seem to have cut off the vanadium peak, but we could see that there was vanadium in this, and that's how we determined what it was. The other thing we did is we took some of the vanadium coating on moon milk, extracted the DNA and sequenced that. And it turns out these microbes are, they eat methane or they eat hydrogen. And so up there, being able to eat minerals is absolutely critical to your existence. So this was really cool to find out how different life was way up on the slopes of Mauna Loa. So now we're going to turn to the work we're now doing at Lava Beds National Monument. This is a NASA project. My husband's a cave photographer, and so he had them spell out NASA as he took the picture. This is one of my favorite pictures. This is funded by NASA to Jen Blank, who you see here on the end. She's the PI for our grant from NASA. And we're doing two things. We're looking at the microbes, and then she works with a robotic team from NASA to see if they can transform what we find into figuring out, can the rover identify microbes on the wall? So here's the problem. If you're a rover and you're trundling down the passages of the cave, how do you know where to stop? How would you know to zero in on this crack in the wall, look closer at it, and see the fact that there are actually little polyps on the wall, just like you might get if you had colon cancer. Actually, I think you can almost see it. Can't see it very well in either, but they're little tiny black polyps here. And those are one of the things that we found quite a bit of life in. So we made basically questions for this grant. We're looking at these secondary mineral deposits in the lava caves at lava beds, and do they have evidence of life or formal life that could be used to help us figure out where to look for life on extraterrestrial bodies? And is this uniform, or is there sort of a continuum across the cave of mineral features moving to minor microbial content to more major content? And so you'll see that the lava bed sample is a little different. So one of the things we did, and one of the reasons we did, is my husband kept saying, so what's on the bare rock? You got to know what your baseline is. What's on that bare rock? And also it's hard to find bare rock. So we actually took samples of the bare rock, but we looked at the coralloids. We also tested the mineral crusts. And in lava beds, we have the polyps, we have fingers, and we have a new thing. We have the cauliflowers, sort of like little heads of cauliflower. We have the microbial mats, and we have the ooze. So we looked across all of these different ones. And one of the things is bare rock is not bare. So there is a small amount of DNA even in there. This is from 7 samples versus the coralloids. We had 6 samples versus mineral is 11, etc. And one of the things we found is as we went along, there's more and more DNA. And so the things that are obviously microbial have a whole lot more DNA, but these mineral morphologies have enough DNA to tell us a lot about the life that's there. So we're going to look at some of this, and you can see how this compares with what you saw in Hawaii. So here's some of the polyps right here. You can see a little right in here, and this is a place where I unfortunately didn't have Kenneth, or where you'd be able to see it better. And when you look with a scanning electron micrograph, you see all those wonderful filaments. and also some other round forms, which are microbial shapes. When you look at the white knobs on the wall here, notice how these have a nice film on them, and the microbes are poking up through it. I actually think there's sort of a fight going on between the microbes and this silica film, which is sort of growing all the time and covering them. And then the microbes fight their way back out. That was a fun one to find. Here's one of those cauliflowers that we have in the caves there. And you can see a whole bunch of microbial colonies here. Lots of filament is a very common, and then there's a lot of that biofilm that I told you about. So let's see. So can you wait, please? See if I can make it a little quieter. He has a better setup for making it quiet than I do. So these are microbial colonies all over that wall. So this is yellow colonies. So you're looking truly at billions, trillions of microbial cells. These I love because when you look inside them, see how different they look than the other ones? These are sort of fuzzy filaments. And when you look at them closely, you'll see there are little round microbes on these fuzzy filaments. And then if you look real closely right here and down here and up here, these are what we call beads on a string. These look like a woman's necklace of pearls with spaces in between the pearls. But just look at the plethora, how many different shapes there are of microbes. So that's one of the things we see in these is there's just a whole diversity of microbes. This is some of the ooze from the back of Valentine Cave in lava beds. It's got different shapes. It's got a very different shaped filament, and it's got fuzzy filaments, but they're shorter and knobbier. So this one has entirely different shapes. Shape is not a way you tell microbes apart, but you can tell there are different microbes here. So what's the microbial diversity in mineral versus mass? So I've shown you that just from morphology, it looks like there is a difference. So Joe Medley took all those SEMs that we did across this whole continuum, and he counted the number of different morphologies we saw. And so one of the things that you'll see here is that those smooth filaments were the most abundant, and the reticulated filaments were probably one of the least abundant along with the rods. But the microbial mats had the most diversity of shape, and there's a lot of biofilm in them. So we can sort of look across samples at how different they are. Okay, and then we also looked at who's present. And what I want to show you here is, again, look how many nitrogen there are. Like nitro, 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 nitro. And notice the word Candidatus. These are ones that are just in the act of being described. So we only know a little bit about these. But what I've found so far from other studies is that most of the nitro ones are also ammonia oxidizers. So we're discovering that there's something major going on with ammonia or ammonium, which is not something you tend to think of being present in lava caves. So this is a big mystery that we have to look into more. And we've been looking across the whole nitrogen cycle, because in where there's oxygen versus where there's no oxygen, the top half is called nitrification. And you see this done by plants on the surface, but it's where all these organisms take ammonia and convert it to nitrite. These organisms convert the nitrite to nitrate. And then we have some that take that nitrate and transform it back into nitrite. and the cycle continues. So we're looking at how this cycle of life could support the rest of the ecosystem. We also worked with the NASA team. This was their first rover. You may be kind of scandalized by how big it is in terms of moving through a cave if you've been in a cave. But they now have a smaller, really sleek robot that can walk on legs. It looks like sort of a giant insect walking through the cave. And we were able to go in with that team, and we did a ground truthing experiment where we were given a timed amount of time to go along the wall and say, this is a good candidate for sampling, this is a good candidate. Those are marked with these sterilized ping pong balls, so I could say where to go, and they would map them in, and then they would compare that to what the rover would find. And I have not yet heard the results of that, but this was really fun. So what have we found across all of this? So we found that those secondary minerals are actually a good place to look, because it's much more likely that on a planet like Mars or on the moon, in a lava cave, it's more likely we're going to see these secondary minerals. They've got DNA, they've got evidence of past life, even if the things are no longer alive. We're also finding that this phyla of microorganisms, the archaea, may be a really critical part of the community for supplying the nutrients that are needed. And a lot of it is tied up in the cycling of nitrogen, which gives energy. I'm sure you don't think about having a nice plate of nitrite or nitrate for breakfast, but the microbes do. And it has a lot of implications for how we can help NASA scientists and engineers look for life on the other planet. So we're still in the middle of doing a lot of this work. We're going to take the geochemistry work from a different part of the team. Another part of the team is using the lipids present in the microbes. So your fat, for instance, would be a lipid. And we use that to tell whether they also fix the carbon dioxide in the atmosphere, converting it to organic carbon, which other microbes can eat, just like you eat organic carbon. And then we're also doing something called metagenomics, where you can take a sample, extract all the DNA, and you sequence every gene, and you see what this organism is capable of doing. It tells you capabilities, not what it's actually doing, but it gives you a good idea where to look. And so we're going to try to use that to figure out, do they eat organic carbon or do they eat rock? Are they able to transform carbon dioxide into organic carbon? And then we'll also, one of the things I've discovered in doing work like this is it's really important to be willing to work across different disciplines, because within your own discipline, you just don't have the expertise to get the whole picture. So I like working with the geochemists and the engineers. So I want to acknowledge the money that we've gotten from the NASA P-STAR, and Jen Blank as the PI. Joe Medley, for those of you who I'm sure have had students or children, Joe, as an undergraduate student, went out and got $10,000 worth of grants to do the research he did in Hawaii. So he paid his own way to Hawaii. My husband, Kenneth Ingham, did a lot of the photos that you see there, and all these parks, Lava Beds National Monument, and the Cave Conservancy of Hawaii, and the El Mapais National Monument gave us permits and oftentimes funding. And then the landowners on the private lands supported us in doing this. So with that, I will take your questions.
Okay, I see that there are a lot of questions, and Stephen, you've got a list of them. So can you take it away?
I do. I'll just go in order of when they came in. When you're talking about the blue-green deposits, there's a question about, are you sequencing the whole thing?
So that's a great question. So for the work I do to identify who's present, I'm actually sequencing one gene. I know that sounds pathetic, but it's a fingerprinting gene, and it's the one that's used to identify who's present. The metagenomics I referred to is what we've done it on 3 samples from Hawaii, the Joint Genome Institute. When I told them you have no cave samples, did 3 of them for free. And so when they do the metagenomics, they sequence everything. Does that answer the question?
I think so. She can ask a follow-up if she likes, or that person can. The other question is, what about fungi in caves?
Okay. So I also work on a project called, have you heard of white nose syndrome in bats?
Sure.
Okay, so what I've been doing for the last 10 years is sequencing the fungi that live on the bats. And then more recently, we did a small study where we looked at what was on the walls, because what we wanted to know is, are there bacteria that could be a natural defense, and also with fungi. And so we're now analyzing the fungal data to see, because there are fungi that do produce some antibiotics or some ways to control other fungi. And so that's the kind of fungi work that I'm doing, is mainly with the bat. I've done a very small amount on the other fungi, but generally I stick to the bacteria and the archaea. The fungi are not easy to do. And I have personal experience with a fungus that causes histoplasmosis. So they aren't my favorite organisms, because I have sealed off spores or in my heart that will unwall when I'm old and have a weak immune system and kill me. So they're not my favorite organism. Even if it was my own fault, I got the spores.
Wow. Another question is, will the current rover on Mars be looking for microbes, and how will it do that? And then,
Okay, so that's a great question. So there is an article that just came out in 2020 called, it's Mars Extant. And I can give Jill or Stephen, if I can, I can put it in the chat. We just published our report, and it has sort of some of the plan for how we're going to look for life on Mars and its microbial life we're looking for. Now, these rovers are equipped with various sensors that scan for chemical compounds that are indicative of that life might be there. I don't know if the rover we used had the ability to look for methane, but there's a woman, Carol Stoker, who like 10 years or more ago found evidence that there might be methane in the atmosphere of Mars, and there was a more recent discovery of that. So a lot of it is looking at that kind of thing. There's a lot we still need to do to make the robots better at doing this, but NASA's doing a whole lot of work on that. Let me see if there's any possibility that I can put this article.
okay.
Let's see.
While you're doing that, there's a related question. What does the existence of microbial life indicate for the existence of other life forms?
Wait, what does the existence of microbes say?
Microbial life indicate for the existence of other life forms?
It's more what they can do for the other life forms. It doesn't say one way or another whether the others are present, unless you're actually looking at ones of those on those organisms. So I don't know that that really tells you that much. But they do a lot for those other organisms. And I used to work on invertebrates in caves. And so we use other techniques to look for them. But I can tell you some of those invertebrates also have microbial symbionts, things that live within them.
Okay, one quick interruption, if I may. Diana, Phyllis Friello is on the chat, and she works for NASA in Houston, and she has some information about the rover, I think, right, Phyllis?
Great. Would you add that?
Can you unmute Phyllis and give us a quick?
Sure. So for the latest rover, Perseverance, that landed on February 18th of this year, they've got two really cool instruments, Pixel and Sherlock. And Pixel will be looking for those remnants of the biofilm, and Sherlock will be looking for the organic compounds. So they're both up there now, looking for these different components. And then with that, they have Watson. And I love the guys who named these things because they're so cool. So when the instruments go over, Pixel and Sherlock, Watson will also be taking photographs. And then in addition to that, they will be drilling down just a little bit into the soil, into the regolith, and taking samples to cache them for hopefully future pickup.
Okay. Do you know if when they detect those carbon compounds, can they tell what the compound is?
They should be able to, yeah.
Okay.
Once they detect them.
Because there are some that would be much more indicative of life. So yeah, that's exactly, thank you. That's very helpful.
Should be pretty cool.
Yeah.
Thanks, Phyllis.
I'm looking for there being a scanning electron microscope on the next rover.
That would be so cool.
That's what I want. Can they detect ATP?
I'm not really sure about that. I don't know what they would use to detect that up there, what they have on board. Curiosity might have something, but I'm not really sure if they could do that.
Yeah.
I know that one thing that came out recently that was really cool, that PAHs, polycyclic aromatic hydrocarbons, were just detected in a cluster out beyond, but that was, they thought they were out there, but that was the first time. So who knows?
Okay. Yeah, Penny Boston would probably know more about that too. I did put the article by Brandy Carrier et al. This is the result of a conference we did last year and came up with suggestions for looking for life on Mars. So you might be interested in just scanning that to see what's there.
Great. We have one other question. You know, I hear that there's an echo in my voice. I apologize for that. Someone's asking, could the ammonia in its caves be excretion or waste?
It is possible, and we also, as part of the study at lava beds, looked at the invertebrates that are present. And I didn't show you any pictures, but the other thing we find on the walls of the lava caves are diatoms and what are called testate amoeba. And then one of the people on the team is sequencing all the eukaryotes, so the higher level organisms. And so she's been able to show that there are lots of other things that come in the cave. And having studied invertebrates in the caves, I know that there are a lot of small insects or arthropods that use caves as a home and live on the walls. And so there are also pack rats. So we saw pack rat middens, and I'm sure the pack rat middens are full of ammonium. And so that, actually, you're right, that could be a source of the ammonia, because there's could be evidence in some of these caves of the pack rats. So yeah, we should look at their records, and a few of the cave have bats in it. And you may know that if the cave has a lot of bats, like Bracken Cave in Texas, the ammonia levels in the air are so high, it would do very bad things to you. Anybody been in a bat cave?
Yes.
Two people, Jill and Loretta.
And Donnie. He's got his thumb up.
Ah, Donnie. Yeah, I've had rabies shots many times now.
Yikes. Are there any more questions, Stephen?
No, this is it for now.
Okay, does
Anything
any,
that occurs to anybody?
Yeah, anybody else got a question? How sensitive to the temperature and humidity? How reliant, I think that means. Is that what he said? Thomas, is that what you're saying? Where are you?
How resilient, how easily can they die if you have changes in the environment?
So if it's an invertebrate, really easily. So the camel crickets that I have worked with for many years, yeah, you have to be, they come out at night when it's more humid. They do come out of the cave. But the microbes, I actually did like tests on temperature. And one of the things that always just makes me really grumpy is people often will try to culture the microbes from the caves, and they will put them at 37 degrees. That kills cave microbes. So they are in like some of the caves in New Mexico, the lava caves are -2 centigrade in this one cave, up to 9 degrees centigrade. So they live at that temperature their entire lives. So they are adapted to a narrower range of temperature. So temperature certainly will. I haven't with microbes seen that humidity makes as big of difference, but I bet with invertebrates it would. Because yeah, that's a very good question, because that's a problem for growing these. And so we grow them. We have one incubator. We have a place that's 4 degrees centigrade, an 8 degrees centigrade, and a 20 degrees centigrade incubator. And we try to, get things pretty quickly into those. Microbes are resilient, but there's a limit to their resilience.
Thank you.
Yeah.
Okay, does anybody else have another question for Diana?
Or comments?
Or comments? Yes. Well, a virtual clapping, I don't know how we do this, but if we all kind of go like this. Yes. Thank you
all.
Thank
This
you
is fun
so
to
much,
do.
Diana. And I would like to,
appreciate your interest.
well, it's the International Year of Caves and Karst, or Karst and Caves. And so I would like to get as many cave talks as possible this year, because it's just fascinating and we need to have more information about caves to the general public. But very quickly, for those of you that are still here, the next month on the second Sunday in May, we have a talk about climate change and how that affects birds by a local ornithologist here in town named Tom Tribble. And then in June, Phyllis, who just answered some of the questions and made comments, who works with NASA, she'll be talking to us about life in the space station. So
that
please,
sounds neat. Send me a link for that one.
I will, and please stay tuned, everybody, for the meetup announcements. And thank you so much for joining us. And Diana, again, thank you. It was really interesting. And someday I'd like to we'll go caving in Hawaii together.
That would be fun. We spend 1 or 2 months a year over there.
Perfect. I'll be there.
Which is really fun to do. And it's way easier than Lechuguilla.
Oh, okay. All right. Well, thank you everybody for joining us, and we'll hopefully see you next month. And bye. Thanks.